Dr. Jeff Rubens from Johns Hopkins University. And Dr. Rubens is the winner of the 2016 Giant Next Gen Award, actually. So congratulations for that, too.
Thank you. Thank you for having me here today. I'm very excited to share my research with all of you. So I'm going to talk about targeting mTORC2 to enhance cisplatin-induced cytotoxicity in ATRT. No financial disclosure. So atypical teratoid rhabdoid tumors are the most common malignant brain tumor of infancy. These tumors are treated with a cisplatin-intensive regimen of chemotherapy, and tumors tend to initially respond very nicely to therapy. Unfortunately, this response is very short-lived. Tumors gain resistance and very quickly start to grow again. And this leads to a median survival of just 6 to 11 months. So we're really in dire need of developing new targeted therapies to improve survivals in ATRT. This aggressive phenotype is born out of just one recurring genetic mutation. So there's a mutation in the SMARCB1 gene. This gene is part of a SWI/SNF chromatin remodeling complex. Dr. Brown gave us a nice explanation of the epigenetics of tumors, but what this complex does is that it unwinds DNA to allow for the transcription of many different genes, so that when you have a mutation in SMARCB1, you have an altered SWI/SNF chromatin remodeling complex, which then leads to a transformed or abnormally regulated epigenetic landscape in these tumors. And this can lead to the overregulation of many oncogenic pathways that then can lead to this aggressive growth in ATRTs. So in our lab, we're looking at what are these overregulated, upregulated pathways in ATRT that's contributing to this growth of the tumor, and then trying to figure out how we could link that to pharmacologic inhibitors that would then reduce the tumor genicity and improve survival. When I joined the lab a few years ago, Dr. Rob, who we just heard from, was just finished up on a project where he identified very high levels of Lin28A, a stem cell factor in ATRTs that appeared to be contributing to this tumor genicity. It was one of these upregulated pathways that really was driving the growth of these tumors. So what he had done, as you can see in the Western blot here, is that he used short hairpin knockdowns to knock down Lin28A in ATRT. When he did that and he grew the cells in culture, he found that the knockdown cell lines grew much more slowly than the wild types, as you can see in the MTS assay here. He also showed that it upregulated apoptosis, and then when you put these cells into xenograph models of ATRTs, the knockdown of Lin28A, as you can see in the red bar here on the Kaplan-Meier curve, extended survival from about a median of 50 days to over 100 days. So it appeared that Lin28A was really one of these pathways that was being upregulated in ATRTs and contributing to the strong growth in ATRT. Unfortunately, there's no inhibitors of Lin28A. There's no drugs that we could then treat our kids with ATRT to improve survival. So where I came in was trying to figure out what was downstream or what was being regulated by Lin28A that was really responsible for this aggressive growth. And so I turned to the primary literature and tried to figure out what Lin28 was regulating. And it appears in other tumors that Lin28 could be regulating the mTOR pathway. We know that mTOR is upregulated in many aggressive cancers. It leads to drug-resistant and poor survival in many aggressive cancers. And so we decided to look further into this pathway. mTOR really works through two different pathways. It has the mTORC1, which phosphorylates the S6 ribosomal subunit that leads to a controlled cell growth, protein synthesis, and metabolism. There's also a TORC2 complex that you could see phosphorylates AKT and could further regulate growth in ATRT. Less is known about the TORC2 complex. So we initially looked back at these Lin28A knockdown models to see how it was impacting mTOR expression in ATRT. We found that when we knocked down Lin28A, as you can see in each of these short hairpin models here, we had a subsequent knockdown in both TORC2 expression, as you can see from phospho-AKT, as well as TORC1, as you can see from phospho-S6. So this made us believe that potentially, as we had seen with Lin28A, that the mTOR pathway may also be highly activated in ATRT. So we worked with our collaborators at St. Jude. Brent Orr had created a tissue microarray where he had 18 primary ATRT models where we could then stain for activation of the mTOR pathway. What we found when we looked for TORC2 expression is it really had intense and broad activation in ATRT. So as you can see here, 60% of our tumors had this very intense pattern of activation, and all of the ATRTs had some activation of TORC2. TORC1 was also activated. You could see that there was some tumors with a fairly intense activation of TORC1, but not nearly to the degree that we saw with TORC2. So we know that there's medications like everolimus and rapamycin that are on the market that are really good inhibitors of TORC1. But we felt with TORC2, with ATR2, we really needed to inhibit TORC2 as well to fully inhibit this pathway. So we turned to the medication TAK-228. This is a dual TORC1, TORC2 kinase inhibitor. It's been in phase 1 clinical trials. It's shown that it's safe. It crosses the blood-brain barrier nicely. And conveniently, it's also available through the Cancer Therapeutics Evaluation Program. So we found in our ATRT cell models at very low concentrations of this medication, just 10 nanomolar concentrations, it's really sufficient to inhibit our target. So we're inhibiting both TORC2, so you can see here, and TORC1 at 10 nanomolar concentrations. And at the same concentration, when we did MTS assays to look at the growth of ATRT, we're slowing ATRT growth, as you can see in the red line here on our MTS assay. We also completed cleaved caspase-3 assays where we treated tumor cells for 72 hours, and we looked at the induction of apoptosis. So all of the cells are stained with DAPI staining, so you can see all their nuclei in blue here, and then the cells that are undergoing apoptosis are highlighted in red. That's staining for cleaved caspase-3. We found that with the treatment of TAK-228, we significantly induced the apoptosis at the 10 nanomolar concentration, and then when we increased that to 100 nanomolar, we further enhanced this induction of apoptosis. Finally, we created xenograph mouse models of ATRT, and we treated with 1 milligram per kilogram of TAK-228. This is equivalent to the maximum tolerated dose found in adults in human clinical trials. And we found that we significantly extended survival in our ATRT models. So you can see with our BT12 xenograph mouse model, we more than doubled survival in ATRT. CHLA-06 is a very aggressive cell line. It kills mice very quickly, but we're still able to significantly extend survival with this single agent. So we're very excited about these results. But as Dr. Tereski had mentioned earlier, there isn't a very good track record of a single agent targeted therapy really having profound impact on survival in humans. So we wanted to take these results, we wanted to figure out how could we enhance the survival effect by combining it with other medications. A couple other things that we found in here that were interesting. So when we euthanized these mice at the end of the experiment, we were able to dissect out the tumor, and we were able to look for the expression patterns of our targets. So we found that our medication was in fact crossing the blood-brain barrier, getting into the tumor in sufficient concentrations to inhibit our targets. So you can see that both TORC2 is inhibited, TORC1 is inhibited, even at the time of the death of these animals. So this both told us that we're getting our medication in sufficient concentrations, but also that the tumors were finding a way to get around mTOR inhibition to further grow and end up killing our mouse models. So I told you at the beginning that the standard therapies for ATRT is really the cisplatin-intensive chemotherapy. So we were wondering if maybe TAK-228 could combine with cisplatin to further improve survival in ATRT. So when we combined it in our cell culture, we found that the growth of the cells were further slowed when you combine TAK-228 with cisplatin, as you can see from the MTS assays. What was really exciting is that it really enhanced the cisplatin-induced apoptosis. So you can see in our cleaved caspase-3 assay down here cisplatin by itself could induce apoptosis in about 20% of cells, but when you combine it with a low concentration of TAC228, that's enhanced to nearly 35% or 30% here. And then when we created our xenograph mouse models and we combined these medications, we saw a really nice improvement in survival. So again, we're using fairly low doses of cisplatin, and at those concentrations, it really had no impact on survival by itself. TAC228, again, is showing that nice improvement in survival. But when we combine the cisplatin with TAC228, we see a further enhancement in survival, and then this population of mice that are able to survive significantly longer. So we next wanted to see what was happening. How are these medications combining to have this greater effect? So we had seen that TAC228 and cisplatin further induce apoptosis. We thought that TORC2 was probably responsible for this effect since it was much more broadly intensely activated in ATRT. So to look further into how TORC2 impacted cisplatin-induced apoptosis, we forced the expression of AKT, or forced the expression of TORC2, as you see we successfully did here. We then treated these cell lines with cisplatin and found that when we do increase TORC2 expression, we really protect these cells from cisplatin-induced apoptosis. Similarly, we see the same effects in the cleaved caspase 3 assay. We then took cell line that had very high level of activation of TORC2 at baseline, and we decreased that activation using an siRNA directed at Richter. You can see, again, we were successful at decreasing the activation of TORC2. And then when we treated these cells with cisplatin, we were able to greatly enhance the effect of cisplatin on apoptosis, as seen through our cleaved PARP Western blot here. So we're able to determine that really, if we're able to eliminate or inhibit mTORC2 activation, we're able to make these cells much more sensitive to cisplatin therapy. Then we were trying to figure out exactly how this could be happening, and we looked at the BCL2 family of proteins. So BCL2 has pro-apoptotic and anti-apoptotic proteins that are really balanced to try to control levels of apoptosis in the cell. And at baseline, you could see that proteins like BAD, BAX, and BAC really balance out these anti-apoptotic proteins, BCL2, MCL1, and BCLXL. So we thought maybe TAC228 somehow was shifting that balance of apoptotic proteins to really push in the favor of the pro-apoptotic proteins. We're able to find just that. So when we treated these cells with TAC228 for 24 hours and looked at MCL1, one of the anti-apoptotic proteins, we see that we really decreased the expression of MCL1, both with TAC228 by itself and then in the combination with cisplatin. However, we did not see we actually found that the treatment with TAC228 reflexively increased some of these other anti-apoptotic proteins. So especially in combination, you see a reflexive increase in BCXL, BCL2. So rather than, again, this huge increase or weight of the pro-apoptotic proteins, we may have just slightly shifted cells in the balance of the pro-apoptotic proteins like this. So we wanted to see next if we could further enhance the or decrease anti-apoptotic proteins, really shift the balance to the pro-apoptotic proteins by adding a further medication. So we looked at navitoclax, which is a small molecular inhibitor of BCL2 and BCLXL. This medication has also been in phase 1 clinical trials in adults. It's been shown to be fairly well tolerated and cross the blood-brain barrier to get into the CNS. And this conveniently is also available through the CTEP program. We found that when we treat ATRT cells with both of these medications, we're not just inhibiting MCL1 as we see with the TAC228 treatment, but we're also decreasing BCL2 and BCLXL. You see, again, navitoclax causes a reflexive increase in MCL1, but with the combination of all three, you're really decreasing expression of all these anti-apoptotic proteins. So here we feel like we're really shifting the balance strongly in favor of the pro-apoptotic proteins, which may further enhance the effect of cisplatin and decrease that drug resistance that we see in ATRT. And when we do treat these cells in combination, we see that the combination of the two medications slows cell growth compared to each treatment alone. And then when we look at induction of apoptosis. We take this level of about 30% apoptosis with TAC228 and cisplatin alone, and we push that up to nearly 50% of the cells when we add navitoclax. So, in conclusion, I've shown you that TAC228 by itself improves survival in ATRT. The survival effect is enhanced when it's combined with cisplatin treatment. TAC228 is able to enhance cisplatin-induced cytotoxicity through the inhibition of MCL1, and potentially adding in navitoclax, which inhibits other anti-apoptotic proteins, we may be able to further enhance the survival. From here, we are teaming up with Kathy Warren over at the NIH. She's really an expert in drug development. She's going to help us with pharmacokinetic and pharmacodynamic studies that will help get this medication further developed into phase 1 trials, looking at the tolerance of TAC228 by itself and in combination with cisplatin recurrent brain tumors. And then we'll look at more of the survival benefit of navitoclax in combination. So I really just want to take a moment to thank the Children's Cancer Foundation for the Next Gen Award. It was really instrumental in all of this research, pushing this research towards clinical trials. And personally, the funds have helped me with promotion, and I'm going to join the faculty at Hopkins largely because of that money from CCF. So thank you very much, both from CCF and Giant Food. I want to thank my mentors, Eric Robb and Charles Eberhardt, and my wonderful lab at Johns Hopkins.
Executive privilege here while the microphone goes there. If the mechanism is really through shifting the balance in the apoptosis pathway, then you would expect TAC228 to do the same to other chemotherapeutics that would induce apoptosis. Do you see that, or is there something really specific mechanistically happening down below with cisplatin?
Yeah, no, I think you're right. We use cisplatin because it's such an important part of ATRT treatment right now, but I think likely because it shifts the balance, any sort of cytotoxic chemotherapy probably could be used. So anything that has good brain penetration would be reasonable to test, but we haven't looked at many of the agents yet.
Nice talk, Jeff. I have a couple of questions as it relates to ERK and AKT as downstream effector pathways of RAS. I think I've heard you say in the past that Lin28 is upstream of RAS, and so you're looking at mTORC2 as one of the effector pathways downstream of that becoming activated. And so you're using TAC as an mTOR kinase inhibitor, and therefore that's a drug that's going to hit both mTORC1 and mTORC2. And then looking as your readout at phosphorylation of 473 as your TORC2 readout and phosphorylation of S6 as your TORC1 readout. So I guess I'm curious, I thought that AKT required both 308 and 473 for its activation, so I'm curious if you've looked at what happens to that. I'm curious if these cells respond to rapamycin, if there's if they require C1 at all, and then what happens if you showed really nice data showing that if you knock down RICTOR, you can inhibit the cells, but what happens if you knock down RAPTOR? And then my final question would be, there's a lot of crosstalk between ERK and AKT signaling. And do you know whether there's a role for ERK signaling downstream of activated RAS in these cells, whether there's a feedback reactivation of ERK? Would it be beneficial to inhibit both pathways? Does one go down, one go up sort of situation?
Great. So I'll do my best to remember all the questions, but I think those are fantastic questions. And the first, or it might have been the second one, about mTOR1 inhibition. We didn't look at rapamycin, but we looked at everolimus, which is used commonly in brain tumors, and it really it could slow cell growth. It doesn't cause that enhanced cytotoxicity that we see with cisplatin. So we've looked at cleave-PARP and cleave-classphase 3 assays, and it really keeps it right on the level of what we see with cisplatin. So we think it's really mediated through TORC2 that we're getting this effect. Ideally, we could just have a TORC2 inhibitor. I'm not sure that TORC1 is important in the mechanisms here, but as you know, there's nothing that exists right now. The crosstalk with the ERK pathway, I think, is very interesting. We had looked at trametinib in these tumor cells because, as you said, Lin28A or Lin28 has been described to regulate the ERK pathway as well. We saw some in vitro efficacy of trametinib, but it didn't translate into the in vivo setting, and it might be an issue of getting into the brain in these tumors. I think, with pilocytic astrocytomas, they might be more vascular and might have some better ability for trametinib to get in, but we didn't see the efficacy with ATRTs. We did, and it was unaffected. Yeah.
Okay. So simple question. Why is Lin28A non-druggable?
So that's a good question, and I think there are actually several labs that are looking at figuring out how it could be druggable. There's a lot of Lin28 expression in normal cells and stem cells especially, and so potentially the side effects of inhibiting Lin28 might be too great to be tolerated, but people are pursuing the inhibition of Lin28.
So how could you drug it?
So a good way to drug it would be through its interaction with LET7. So its main function is regulating the LET7 family of microRNAs. So if you could have potentially a dummy LET7 or something that absorbs a lot of Lin28, it might be a good way to drug it.
And lastly, when you showed those changes in the BCL family proteins, was that because of protein degradation or splice shifting to a different isoform?
It's a great question and something we haven't looked into yet, but something we should explore further.